Hi, everybody. My name is Rizwan Chaudhry, and I'm your host for episode 3 of our podcast of Organoids and a Large Portion of Chips, brought to you by Work.Community, and we'll be talking about all things organoids and organoidship. And today, I'm delighted to join by my regular guests and experts, Professor Marianna Prof de Julia, who is Head of Urology Research Laboratory at University of Bern, and Professor Stephen Ray Wilson, Department of Chemistry at University of Oslo. And today we're not joined by one guest, but two special guests. So I'm delighted to introduce that we've got Professor Roland Seiler, the head of urology at the Hospital Centre in Biel, and also Bernhard Kiss, who's the associate professor and deputy chairman of the Clinic of Urology in Bern. And both Roland and Bernhard will give us a quick overview of their background shortly. But before we do that, just to say that our topic today is going to be what have organoids ever done for me or for you as you're listening and watching, translating science in a dish to patient outcomes. But before we do that, Marianna, Stephen, how are you both? I haven't seen you in a while. What's been happening?
Oh, we've been doing great. We have progressing quite a lot. We have a new paper accepted in Oncogene that is on organoids. Soon I will share this link with you guys so you can enjoy also how we can measure weight and density on organoids. And maybe, Steve, this is something you might be interested in at one point to chat about as well. And so, yeah, a lot of cool stuff going on.
Fantastic. And Stephen, how about you?
Well, I have been traveling less, so I've had more energy, I guess, to do institutional responsibilities and so forth. But we also have a new paper, which is I think right around the corner. We sent in our revisions for this for doing sample cleanup for drug and drug metabolism studies for organoids. So pretty happy about that. And yeah, so good. It's getting a little colder in Norway, but that's all right.
Cool. All right. Well, look, let's go on to our guest now. Gentlemen, it's lovely to meet you both, first of all. Now, I know my intro hasn't done any sort of credit to your expertise. So I'd like you to tell viewers a bit about your background. So, Rodney, if you'd like to start, just give us a bit about your background and also what your work involves in terms of organoids as well.
Well, thank you very much for the invitation and for having me and for this nice introduction. My name is Roland Seiler, and I'm chairman, as you said, in the Department of Urology in the Hospital Center in Biel. I did research when I started in pathology, which was about 2008, and became interested in bladder cancer. But maybe one of the most important changes was 2016 when I met Marianna, and we started collaborating actually quite efficient, I think, quite successful. And we started working on bladder cancer organoids with Marianna, of course, and we were able to establish protocols, improve pipelines. And we got further in our analysis of these tumors and ultimately thought our pipeline is actually ready for going into clinical trials. So about a year and a half ago, we started recruitment for a clinical trial using organoids for drug prediction. So this is pretty much where we are. And we try to think to bring organoid experiment back to the patient. So we bring it as close to the patient as possible. And I like to work with Marianna in this manner because it's always patient oriented, and that's lots of fun for me.
Fantastic. And Bernard, same question to you. I mean, what's your background and what's your relationship with organoids?
So I'm a board certified urologist who is in the Department of Urology at the University of Bern since 2008 with roughly 2 years of postdoc in Stanford. And Marianna and we collaborate a lot on basic translational and clinical research questions, and Roland and me as well. So Roland and I did a lot of bladder cancer research together, also some organoid related research, and basically we are a well-established team, I would say.
Fantastic. Right. Perfect people then obviously to talk about translating science in addition to patient outcomes, which is why you're our guests, obviously. So, first question for you though, there's obviously lots in the media, but in reality, is this science fiction or science fact? I mean, what do we mean by dish science helping patients? And do organoid responses to drugs really relate to human responses in therapy? So, Roland, Bernard, I'll throw that out to you two as you guys actively involved in this, and maybe you can tell our listeners a bit more about that, what your views are on it. Who would like to start?
I can start if you'd like to. I think it's a double-edged sword because I think in conventional cytotoxic chemotherapy or antibody-based therapies, drug conjugates, organoids are quite nice and well-established, or more or less well-established research tool. For immunotherapy and immune checkpoint inhibition, it's not so much established. I mean, we work on protocols for that, and Marianna is also spearheading that. But I would say we are not there yet to really go into clinic with immuno-based organoids, I would say.
Right. Can you mind me ask a quick question? What are the challenges in stopping you getting to that point then in that particular area?
There are a myriad of challenges to have a well-functioning immunosystem within an organoid that also recapitulates the individual patient's immunosystem. So that is a really tough research question to answer, I would say.
Right. And Roland, what would you like to add to that, if anything?
Well, I think there is two, at least to my mind, there is, taking out the caveats on immunotherapy, of course, but there is two aspects that I think organoids are very interesting for the patient. And number one is the patient that has nothing to lose. He's end of the line.
Sure.
There is no established treatment anymore, and the only thing that he can get is a drug screen and organoid to get any idea of what could work besides just picking something out of the cupboard. This is one end of the patient. The other end of the patient that we are actually became more interested more recently due to our clinical trial is, can we actually bring it into patient routine early in the disease as an adjuvant treatment, or maybe even earlier on, so that you would treat patient according to the organoid drug screen as part of just as a side to the standard of care. So this is maybe a bit earlier in the disease stage, which I think is also very interesting for the patient because it opens totally new aspects of treatment.
Right. Marianna, Stephen, do you want to add anything?
Yeah, I was wondering about Roland. That's a very interesting concept, and I'm wondering, are there like any ways to sort of like benchmark that type of work so that you can have like a system where you would test if that effort would be valid, and then you could extrapolate that to others. I mean, or is this still very much would this still be very highly experimental from case to case, do you think?
Well, there are some steps to go. Obviously, I think one thing that we can measure response in organoid, that's feasible. But how would you measure response in the patient? And of course, you can use endpoints, but weak endpoints like recurrence-free survival or maybe progression-free, or you do a CT scan and see does the metastasis shrink. But I mean, of course, these are endpoints, but it's not very rigid. So what you would like to have is actually a measurement of response in vivo in the patient. So we are currently also thinking could we do a head-to-head treatment installation in the bladder while the tumor is still there, and using an organoid to do the same and see does the response in the organoid reflect what actually happens in the patient. So measurement of response is critical, I think, to move a step further to know, what's happening in the organoid is also what's happening in the patient.
Cool.
Bernhard, anything to add to that?
No, I think that's a perfectly summarized answer from Roland.
And Marianna, you hunched over your desk looking like you want to say something.
Well, it's more that I completely, I mean, Roland, Bernhard, and we talk about this really extensively. I mean, and one of the also concerns in a way that we have is how do you also really run a clinical trial on something like this? I mean, because, when you submit a trial, and we've gone through this exercise a couple of weeks ago to expand one of the trials that we are running together, a lot of times we get questions like, well, is it a randomized trial? How do you group patients? Do you have a control group? so the whole concept of how a trial could and should be run with organoids as a predicting element for a concept of precision medicine or precision oncology is a different concept than a trial in which you're testing one drug and seeing how this drug works on all of these different patients, on all the patients, because precision medicine means exactly this. We're going to have like 100 drugs and they're going to all work differently. And so how do we make that into something that's actually tangible and reasonable and approvable also?
That is nice. Sorry, would you like something? Add something, Bernhard?
Yeah, I would like to add something. I think the setup for like having an in vivo response or a direct in vivo comparison can only, at this point of time, can only be in a highly palliative setting where you have the patient with fully metastasized disease, and you can monitor the patient via CT scan or whatever, a ctDNA. Directly in the clinic. For the earlier disease stages, I don't see that how that should work from an ethical standpoint to leave the tumor inside the patient if it's potentially curable. So that's a tricky thing.
Well, we'll talk about ethics in a little while, actually, Bernhard. But yeah, anything else anyone would like to add to this particular question?
Not really.
All right. Well, leading on from that, actually, what are the general opinion on organoid co-trials? I mean, what is the opinion about that? I mean, can they really be used? Are they being used? And how does it work?
If I might start again, I think it's a really intriguing idea, and I think co-trials with organoids should have a bigger place in the research community, I would say, as they have now. Because right now, ctDNA is very, very hyped, which is also very nice. But I also think that co-trials with organoids should also be performed more frequently, I would say.
And without sounding stupid, I mean, is there a barrier to why that isn't the case right now?
I think there are not so many labs who have a well-established organoid pipeline. A lot of people claim they are doing good organoid research, but in reality, there are not so many places who are really doing good organoid research. So I think that might be a limiting factor. To get a hand on a really well-functioning organoid core might be a limiting factor, I would say.
Roland, anything to add to that?
Well, I think one of the issues that a lot of research facilities and clinicians maybe also face, and this is probably what we overcame quite successfully, is to bring actually the patient close to the lab, or to bring the clinician close to the scientists and vice versa, that you have a proper sampling, that you have a proper transport of the sample to the lab, and then you have a lab that have a successful pipeline that actually works. So it requires more than just a lab. It also requires more than just a hospital. It actually requires clinicians and scientists that work together and then communicate together. And this brings then the possibility to do such trials that I think are very important for the patient, but also for the system itself, for the organoid system itself. I mean, in treatments of infections and using antibiotics, we use this model for a long time. I mean, we do culturing, we do testing of sensitivity, and then use the antibiotic that is most sensitive. So this is no question. So why should this not be at some point similar used in cancer? It's more complicated, obviously, but I think it's a way to go.
Can I just add, I think that one of the things that maybe we should stress a little bit more is the fact that the kind of collaboration that we have created among the three of us also implies an institute of pathology, a shipping company, people in the lab. I mean, it is a really, so we describe it as a pipeline, but it is really something that took really a lot of effort and communication. And also the advantage we have is that both Bernhard and Roland are actually surgeons. So they are actually the ones that are sampling the material, because you know, if you sample a material and the material is not of good quality, what we get out of the lab will not be of good quality. And people like them, they really put an effort in what they do and try to really communicate with us. So we went through a process of first of optimization of what kind of sample we needed for us to make sure that we could actually do this. So it's so although all our protocols are mostly published, there's a really deep background of know-how that is really that is important, and most importantly, there's this willingness of everybody to try to do this.
Right.
And that's actually one of the points that I wanted to raise. You asked what was one of the limiting factor in why are these organoid, why is not everybody doing this? Well, there's also a lot of unwillingness and how what is the right word that I want to use? And a bit like of skepticism from the medical community, I would say,
Right.
that goes towards, well, is this something that can we really do this? And the question that we usually get as researchers is, well, did you run the clinical trial? So, and it's like catch 22 or 23 or whatever it is. So to, you know, said, you know, and so, but I think that there are people out there that are interested, and we just have to find them. I think it's like.
If I might add something, and I think Marianna really raises a very important point. Roland and I, we impersonate like the concept of a surgeon scientist. We both did our time in a research laboratory, and we know how tedious it is to get really a nice piece of organoid or a nice piece of research tissue out of a surgical specimen. And I want to advocate the concept of surgeon scientist a little bit more in clinical trials like that, because it is a rare species to have basically people who know both sides of who know the bed and the bench. So
Sure.
that's important in trials like that, and setting up a pipeline like that. And Marianna and Roland really did an amazing job in their phase 2 feasibility trial they set up. So that is really to be appreciated.
Brilliant. Okay, well that's good. Now let's. Sorry, you're going to say something. Go on.
I need to just ask quickly. It was also discussed by Bernhard also and the rest of you, like, you know, the quality of the models, like the organoids, that some labs may be doing better or worse organoid models. And just as a simple analytical chemist, which I am, because I would like typically evaluate another analytical methodology from, for example, how has it been validated? So you would like follow FDA guidelines for the quantitative and the qualitative measures. And that this can be like for small drugs, but when you get up to like metabolomics or proteomics, things get a little bit more murky in how things are validated. But still, the question is, how would you guys assess, or if you see somebody, what would you say is a good organoid model? So if you were to see a paper or work from another group, what type of criteria would you say that, okay, these guys seem to be knowing what they're doing, these guys seem to not quite know what they're doing yet, if that's a fair question to ask?
Probably Marianna can respond on that. No?
Yeah, so we're just gonna create a lot of enemies right now.
No, you don't have to name drop anybody.
That's a very difficult question to answer. I mean,
Yeah, sure.
meaning that there's the art of writing a paper and presenting, you know, your data, and then there's the art of reproducing or seeing what is effectively what's working. And we went through this exercise with, you know, many different organoid from many different tissue types. And what we realized is that there are some things that are described out there that are maybe not as precise as they should be, to allow you to actually generate what it is that the paper ultimately claims it can do. Now, in terms of what is a model that makes sense, there's also these two very different lines of thoughts or schools, let's say, on organoids. And one is that you have to have an organoid that has to passage itself indefinitely in a dish, right? To make sure that it does recapitulate or that it can be used. And then there's another school of thought that says, well, we don't really need to do that. We just need to do short-term cultures that are closer to the human tissue itself. So I'm somewhere on, on the fence on both of it, for the reason that I think both models work. It just really depends what is the question you're using. So they're both not invalid or valid per se, but it's just that if you want to do genomic studies, for example, or stem cell studies, or studies that with a CRISPR screen or something, you need to have a stable organoid. But then, the question that I ask myself is, well, but then you have an organoid line, right? So what's the difference with a cell line once you have an organoid line, right? So you always have to take into account there might be genetic drifting or stuff like that. And instead, for the use of organoids in clinical trial, or the use of organoids to try to assess what a drug might do, I believe that the short-term cultures are the ones that work best, because what we do is that we don't allow the drift. We don't allow the cells to change in that short time. I mean, in the case of the trials that we're running for bladder, it takes us 10 days from when the surgeons give us the samples for us to generate the organoids, to do the drug screen, and give them a response. So it's only 10 days. And ultimately, for some kinds of cancer, time is of the essence, because the patient does not have it. So that's why we try to be fast in what we do. But different things. But if I when I read a paper on organoids, I'm always a hypercritic of a lot of things. And the reason is because this is what I do every day. So meaning that, so I know what the difficulties are. So when people tell me, yeah, we passaged the organoids, one organoid in a 96 well plate, I'm like, really? Okay, how did you do that? So it's but I also appreciate the fact that there's a lot of times that they're not organoid experts that are reviewing papers, right? So it's a balance. And I'm not saying that people are not doing things correctly. They're just doing things that sometimes are difficult to reproduce. That's what I'm saying.
Yeah.
I might want to add something here. I think what we also experienced during our journey, the three of us, is things that are probably not even reported on paper. So what is the viability of cells of the sample that you get in the lab? And is the viability higher if you do dissociate in single cells and freeze and then thaw again for further use? Or is it better if you cryopreserve and thaw to use the specimen, then dissociate after thawing, generating organs? So things that are probably never reported in papers. But we experienced that during our journey, and we tested different methods of sampling, different methods of protocols of processing and of freezing and thawing. And this is very critical, because the higher the viability, maybe the more viable cells you have that are comparable to what was actually in the tumor.
So would you say that that sometimes people they don't report also enough of the information of what they're doing, and that things can also get lost in that? Just picking up on one of the first parts of your.
I mean, you would probably.
Sorry. Go ahead.
You would probably report we sampled 20 cancers and we were able to establish organoids in 18 cases. So it sounds quite successful, but I mean, what if you only have 50% of cells viable in the specimen? So how close is this organoid then to the tumor when 50% of the cells died during the processing? And probably this is not very important information, but maybe information that should go in a supplementary where you can actually read how well established were these protocols.
Yeah.
Bernhard, you're going to say something.
Yeah, Marianna and I, the funny thing, Marianna and I had the conversation with some lay people last week, and it's like having a cookbook. You have a cookbook, and in the cookbooks, you don't read. There are some cookbooks where you can finish the dish without any problems, and then you have cookbooks where you cannot get the dish out of the recipe you actually want to have. And that is probably exactly the same in reporting on growing organoids. I see.
I think that's also a general problem for many sciences. I mean, even for, synthesis, drug synthesis, when people have been reporting how a drug is synthesized, there's also been an incredibly frustrating lack of reproducibility in this, because there are small parts of the recipe, that have not been described properly. It might even be just that there have been a touch of water present or not present that has made papers even be on the verge of being retracted because they just simply haven't worked. And I don't know, I just wish that people would be more willing to put more than less, at least like you say in the supplementary material, because we don't have the problems anymore of limited space in physical journals. So I think that more is more in these cases now.
I agree.
But I think it's more the problem that if you deal with that stuff every day, you don't even think about reporting it, because it comes naturally to you that you have to be aware of certain things. You don't even think about that other people don't know about these things. I think that could
That's a good point.
Now, before we go to the next question, Roland, I know you have to go.
Yes.
So I want to give you the get out now before we start the next question. So
Thank you very much for having me.
I just want to say thank you very much for taking time out to speak to us today. It's been really lovely to meet you, and thank you for your contribution today.
Thank you, and have a great day.
Thank
you,
you,
Roland.
Roland.
Thanks
Take
a lot.
care.
Bye.
Bye-bye.
So as we say goodbye to Roland, my next question leads on based on something Bernhard said a bit earlier, actually, because you talked about ethics and clinical trials relating to organoids. So what are the ethical concerns, in relation to using organoids in clinical trials?
I mean, I see one big ethical concern is what do you do in patients where you don't have a proper response in the organoid? How do you treat the patient? Or do you withhold him or the patient at the standard of care procedure if you have a good response in another drug? So that's a tricky answer from an ethical standpoint, I think.
Right. Anybody want to add anything to that? No? Right.
I think this has been, he really, hit it on the head. I think this is a very important thing. And this is actually, I think, something that is, I don't even think we have any kind of regulations for this right now, guidelines or anything.
Well, let's go one step back as well. How do you actually get a patient sample? What's actually involved in getting a patient sample for a trial or a clinical trial involving
You have
organoids?
to talk to the patient if he or she is okay to get the sample for research purposes. You have to have an IRB in place, like an ethics approval, and you have an established pipeline. You have to have an established pipeline. But the most important thing is the approval of the patient and the IRB approval. So
Right. Okay. And in terms of things like health insurance, the patient's health insurance, is it covered by the patient's health insurance to do things like that? And also, who owns the sample and IP at that point?
I can only speak for Switzerland, and the health insurance doesn't cover. The health insurance does not cover, at least not to my knowledge, the costs of growing an organoid and doing drug testing on it. About the IP, I cannot give you an answer. I'm not aware of who owns the IP of the organoid of a patient. I don't even know what that should be.
So so there's The organoids are not owned as IP, but the material is considered is patient material, and that is owned by the hospital or the university. There are strict agreements between the hospital and the universities with the tech transfer offices that are in place on what can and cannot be done with this material, and also with the material that is biobanked, of course.
Right.
The lists are pseudo-anonymized, meaning that I never know who the patients are, and the clinicians have the key to unlock the codes. So one of the things that we had to do that was very important is that because we don't only sample the tissue, but we also collect blood and urine in the progression also of the disease. Everything has to be anonymized. So it took quite a long time just to decide how these codes had to look like, so that we would not mix all the numbers and the letters up, because that, of course, would be totally unacceptable event to do. So yeah. So
That all,
Roland and Bernhard are the only two that have the codes.
but that also depends on the ethics approval. There are also research questions where you have no idea, nobody knows or can decode the anonymization of the samples, because it's forbidden to have that code. So that depends.
Right.
It depends on the protocol you submitting for ethical approval.
Okay, so going one step further, I mean, if a new drug or procedure process is developed via this, though, who owns the IP? Is that the same situation? You won't know who owns the IP, or is it the hospital or the clinic? Who would own that?
That's still the university hospital and the joint, because we are employees of the hospital and we work under their framework. So it's everything that's developed from us would have, so, and if we are filing for an IP, we would do that via the university. So it's
Right. Okay. Now, looking at that, I mean, Bern, you were talking about some of the challenges a bit earlier. So what steps do we need to take further, sort of developing the clinical trials using organoids? I mean, are there any steps you think are not currently being done that we could do to actually develop them further?
You mean, I mean, of course, money is a reason. It would be fantastic to have we were talking about cold clinical trials earlier. It would be great if organoid testing would be covered by the insurance companies or would be covered by hard money from the university. So that would be one step who would make organoid drug testing more widely used. I think, as in so many things, money is a big issue.
Well, it's funny you should say that, because one of my questions was down the list, if you had $1 billion, or we could use Swiss francs if you like, where would you spend it in terms of this? So in terms of just getting more people to sort of submit organoids for research, or more collaboration between surgeons and researchers, I mean, where would you throw that money then if you had that money in the first place?
Vegas, to make 2 million out of it.
Well, you could blow the whole billion as well in one way as well. So that could work either way, to be honest, Bernard.
Just don't bet on Mike Tyson. That's the only
really not, no.
I think an important point is really to establish immunocompetent, so to say, organoids, and I'm talking about that day in and day out, and Marianna is probably annoyed by that at this point. Especially for bladder cancer, that's a very important point, because the immune system plays such a central role.
Sure.
And I would spend a lot of money to establish a model where you really can recapitulate the patient's individual immune system.
Right. And Marianna, have you got anything to add to that at all?
Yeah. Well,
apart from going to Vegas, you might want to get.
I'll join them in Vegas. Yeah. No, I mean, I think, I mean, Bernard's right. I mean, we are investing some of the research into creating a bladder on chip model, which is the talk that I will give at the meeting in February. So I will talk about this model that we have established, and there we have introduced immune cells. But I always have, so first of all, I'm not an immunologist, and I think that it is very difficult to decide which aspect of the immune system you want to recapitulate in a dish, because the immune system is complicated for
Sure.
a reason, you know. So it's and picking out this cell type or that cell type and putting it in the dish in contact with or without the cancer cells or normal cells or whatever, it's I think it's really challenging. It's a very difficult thing. I believe more that a better maybe approach would be to try to use tissue slices from patients, because they already come with immune cells and the correct immune cells in there. The downside of that is that, well, if you do a treatment that induces, for example, some sort of inflammation and you want to recruit more immune cells, the question is, where are we going to get those from if we have a slice of tissue that already has its own immune cells? So there's a lot of pros and cons, and I think, you know, I mean, we're just not there yet, knowledge wise, I think. But you know, we go, you know, people may surprise us. I mean, I think technology moves really fast in the nowadays, so it's yeah.
Anything else to add to this before we move on?
Just wanted to scoot back 1 second about when it came to the stuff about patents and IP, and it's just a kind of a comment, maybe a bit on the side, but I think this is a field that's, you know, evolving pretty rapidly, and it's quite multidisciplinary. There's a lot of, you know, uncharted territory, and I also think that this is something that will also be difficult for the TTOs also to be able to handle. And I mean, if you look at it historically and scientifically, scientific history from the last couple of decades, you can see that there's been several instances where the universities or the TTOs have not been quite on the ball, so to speak.
Stephen, can I stop you? So what do you mean by TT? Forgive me, as a layperson.
Oh yeah,
What is TT?
technology transfer office. So,
Right.
so this will be the people at, for example, the university who will who are in charge of making a product or a license or a patent, or, you know, administrating the process towards something that can be commercialized. So every university, or at least most universities, will have such an office who will then take stuff that has been invented or is being invented at a university, and they will then be responsible for making sure that this, which is owned, the patent will be owned by the university, will be then handled appropriately, perhaps being licensed to a drug company or so forth and so on. But the thing is that there's a lot of times where technology and the paradigms, they go so fast that the TTOs don't necessarily are able to follow up on it. And then this can be troublesome. This happened, for example, with CRISPR-Cas technology, where people, I think, still are in court because they didn't quite figure out who owned what, you know, with these competing groups. And also how you know, modifications of mRNA, how that was handled, which ultimately turned into the Moderna and Pfizer vaccines. So I guess my point is that organoids and organ-on-a-chip research is, you know, kind of exploding in a lot of ways, both technologically, but as we hear now, also in applied sciences. And I just hope that universities and TTOs will be able to be on the ball to make sure that we will be able to protect these things. And I think we as researchers also have to be very aware of how these will then be handled, because there's very little automation in these processes like that.
Right. Well said, actually, Stephen, and a good point. Now, we're not talking about clinical trials, but really, I know you guys, in terms of where you keep up to date with your latest developments, because we've talked about it in the past, but Bernhard, where do you keep tabs on what's happening in terms of latest developments around organoids and the technology that you need to help you?
Marianna, you want to start?
So, well, of course, where do we keep tab? Meaning, you mean what kind of ventures do we look at? What kind of meetings we
L-
go to? What
Or
kind of
well, I think more latest developments in terms around the whole research area of organoids and so on,
yeah, so
and what's available technology-wise as well.
so of course this as we said, this field moves really, really fast. So every week you see new things coming up in at a level of on chip technologies, and also at a level of organoids. And one of the things that really is really fascinating is also the applied spatial and single cell technologies that are now used on these on these different platforms. And I think that the future is actually going in that direction, meaning that spatial transcriptomics, spatial attack sequencing is really what's going to at one point make the difference. So this idea to be able to reconstruct three-dimensionally the tumor, and then to maybe use that information to modify reagents, media, components, or whatever that you need to actually grow an organoid in a dish. That I think is extremely important. And I think that this platform that Miles has really put together, I think is really at the heart of it, because I think that it's a really nice place to go to and to see what the latest are. Because in research engines like PubMed, although they're extremely very informative and we can find a lot of stuff, you still have to know what you're looking for, and I think that instead the platform for the world organoids and organoid chip is really a much better way of having the information you need in one place. So I think that's and I wish people would see this more, because I think the contribution of people is crucial in this. And so yeah, it's my 2 cents.
All
I
right.
would perhaps only add that I personally follow the work of the key opinion leaders in the organoid field very thoroughly, and actively look out for papers from them or their groups.
Right. And Steve?
No, I think also I would also like to express my enthusiasm for platforms like the World Organoid Research Day event and so forth. And I think one of the key parts of that is being actually able to interact scientist to scientist, and not just, I mean, of course, papers are unspeakably invaluable, but having that ability to, or that possibility to speak to each other and also talk about the nitty-gritty and maybe what is frustrating you in the lab, and maybe getting those little tips that we were talking about before that maybe you don't think about when you write it, but then it comes to you when you're talking. I think that's extremely important. And another thing I think is very important with such types of events is also making sure that it's going to be have some degree of multidisciplinarity in it, because I think that sometimes you can have events, for example, in microfluidics or analytical chemistry or whatever, and a lot of the time it will be kind of preaching to the choir. And I think that if we have from different people who are using the tools and methods from different approaches, there will be a bit more attention and tension and challenging how this stuff is done. So I think that the human element of organoids and portions of chips is a very important factor in this. Yeah.
Good. Well, leading on from that, is there any research that you're particularly excited about? Obviously, apart from your own, Marianna, which I know you're going to publish. So, but apart from that, is there any research that you've seen recently which you're quite excited about? And is there any promising science that's taking place right now that you would like to share?
Well, there is. I was at at this organoid meeting in Rome a month and a half ago, and there's quite a lot of interesting data that's coming out of the labs in a couple of labs in northern northern Italy. And what they're doing is that really investing quite a lot of money and time in spatial omics. And I think that that's something that I think is extremely interesting. And I'm really fascinated by it also because there's this component of the spatialness and the distance, and that is something that I think plays a major role in I can't really point my finger on it yet. You know, I have kind of a feeling that it's how things are organized three-dimensionally, of course, is crucial, not only for how they work, but also on how they respond to drugs, you know, when they're there. Like if you think about a cancer, like for example, pancreatic cancer, you know, it's really amazing. It's of course it's a deadly and devastating disease, but the biology of the cancer is extremely fascinating, because what you have is a situation in which you have a huge amount of stroma that's there, and few cancer cells that are spread like around, and these few cancer cells, you know, manage to be so deadly that the 1 year survival is extremely low in patients. And then the question would be, why? What can we do better? I mean, why are we not reaching those cancer cells with a drug? So it's yeah, I find it very interesting. So it's
Bernhard, anything struck
I
you
think,
recently?
of course, the single cell spatial transcriptomics or spatial omics is extremely interesting. But I think for the main reason why I really think it's very exciting right now is that people appreciate the tumor microenvironment nowadays even more than they did like 10 years ago. Of course, there was like a time where everybody was talking about the tumor microenvironment and the mesenchymal part of the tumor, but the tools were not there to really dive deeper into the TME. So I think exciting times, because we now have very powerful tools to make very nice analysis of the tumor microenvironment, and that is spatially, and that is on single cell level. So that is for me the most exciting part right now of research.
And as I've got you here, actually, one quick question is, I mean, from your perspective, how much do you use AI these days? I mean, and how important is that to you in terms of the work that you do now?
I mean, it's very important. You have so many data, so much data, that it's not possible for the researchers to analyze them without KI anymore. So I think it's not working without anymore.
Right. Okay. And Steve, what about you? What research has struck you or hit you in the face and gone, wow, that's really interesting?
Well, you know, I've been thinking a bit more about the omics approaches, like in proteomics and so forth. We're seeing some quite intensive work being done on achieving higher sensitivity, higher throughput,
Sure.
also down to sort of like single cell analyses. And I would like to highlight stuff that Alexander Ivanov has been doing in Boston, and there are like groups like that that are really pushing the field of what can be done there. I mean, there's also being done more related to tissues and single cell analysis for like more spatial work. This is being done by, for example, the Mount Group and so forth. I think the jury will still be a bit out on what one can do the single cell analyses for, but this will nonetheless be an important aspect for higher sensitivity, but also higher throughput in omics, which I think is desperately needed for it to be a more clinical relevant for big data, also including for AI stuff. Yeah.
Brilliant. Okay. Well, look, before we wrap up, as we do nearly every month, I'm just going to find out what you're doing over the next few months leading up to the end of the year then. So, Marianna, where can people meet you, if anywhere at all, or are you now going to be stuck in the office until the end of the year?
no, I'll be in Switzerland till the end of the year, writing I think my memories. I know that great.
And what about you, Bernhard?
So Marianna and I are working
Help with me.
on a bigger grant together. We are
So you can blow it in Vegas. That's right. Now we know
yeah,
where you're gonna what you're gonna
exactly.
do with that grant.
It is keeping us up at night and until the end of the year, I would say.
All right, fantastic. And Steve, you?
I've been pretty much trying to limit travels. I'm gonna be going to Bergen though in a couple of weeks, so that'll be nice. I'm gonna be an opponent up there. But next year, then it's gonna be loads of stuff going on. So I'm just
We'll save that for episode 4. Don't worry, Steve.
yeah,
We've got to
exactly.
leave some content for the next episode. Come on, let's not cover it
Absolutely.
all. All right. Well, look, I was going to say viewers, well, listeners, I should say, I hope you really enjoyed this episode of organoids and a large portion of chips. If you did, then please like and click and subscribe the button to get this podcast on a regular basis. And also make sure you visit, as Marianna and Steve have mentioned this already, visit the WORC.Community website. You can see it behind on our screensavers behind us to learn more about lots of different things going on in the organoids and organ chip community. There's loads of stuff there. And also, as again, Marianna's mentioned a bit earlier as well, if you haven't already, sign up for Word Plus 2025, which has been held in Cambridge on the 11th and 12th of February 2025. It will be a fantastic event. Marianna and Steve, I think, are both there. I don't want to make
Yep.
sure you are unless you are. I don't know if Bernard's going to be there, but if he isn't, then he's missing out already. And I'll be there as well. So make sure you sign up for that. Link again will be above this podcast. So all that stuff is, first of all, Bernhard, Roland, who's actually gone now, but was a great contributor a bit earlier on, Marianna and Steve, it's lovely to see you all. Thank you very much for taking time out to talk to me today. It's been, as always, as a layperson and non-scientist, it's always really interesting listening to you guys talk about this fascinating, because it is science fiction to me as far as I'm concerned, but I know it's real right now as well. So thank you very much for sharing that, and have a fantastic end to 2024. And listeners, thank you for watching. I hope you have a fantastic end to the year as well, and hopefully you'll hear from us in the new year. So until then, stay well and stay safe. Bye bye.
Bye-bye. Bye.
Bye-bye, everybody.